My lab is working on neurophysiological mechanisms of mammalian hibernation using non-standard animal model, the 13-line ground squirrel. The squirrels hibernate for 5 to 7 months in the cold without access to food and water every year. Hibernation consists of two distinct physiological states, torpor and interbout arousal. During torpor, ground squirrels drastically decrease their temperature, metabolism, heart and breathing rates. During interbout arousal, or IBA, ground squirrels restore their vital signs to active-like state for 24 to 48 hours. IBAs are essential states of hibernation cycle, but its physiological significance is not well understood.
Ground squirrels mate only once a year, in the springtime, shortly after emergence from hibernation. Breeding is preceded by 5 to 7 months of hibernation, during which time squirrels burn over 80% of their fat, surviving hibernation. In other words, they are in a state of extreme negative energy balance.
Reproduction is energetically costly, and a general tenet of life is that most mammals will prioritize growth and survival over reproduction in times of resource scarcity. In line with this, it is generally understood that ground squirrels become sexually mature after emergence from hibernation, when feeding is restored. In this study, we demonstrate that male juvenile ground squirrels initiate sexual maturation during the period of energy loss during hibernation, prior to spring emergence, in opposition to this general tenet of life.
Sexual maturation for males entails the development of functional testes. Testicular growth is stimulated by two important hormones, luteinizing hormone and follicle-stimulating hormone. These hormones are released by the anterior pituitary and travel through the blood to the testes. Luteinizing hormone specifically stimulates testosterone production, which is important for testicular growth. Hormone production and release is initiated by the activity of a group of hypothalamic neurons called kisspeptin neurons.
Juvenile ground squirrels enter hibernation with underdeveloped gonads, low levels of sexual hormones, and reduced activation of hypothalamic brain circuits involved in reproduction. Here, we assayed the physiology of juvenile male ground squirrels during IBAs across hibernation and found increased kisspeptin production in the brain. These findings correlated with increased levels of sex hormones, including luteinizing hormone and testosterone. Lastly, we found that testis size doubled during the hibernation season. Overall, these results suggest to us that sexual maturation in juvenile male ground squirrels is initiated during hibernation despite the negative energy balance state.
Given that sexual maturation begins during hibernation, we wondered whether hibernation itself was necessary for this process. We prevented a cohort of juvenile ground squirrels from hibernating by keeping them at room temperature with ad libitum access to food and water. We observed that these animals initiated sexual maturation, as reflected in increased testicular weight and increased sexual hormone levels, around the same time as their hibernating counterparts. This demonstrated that the timing of sexual maturation is circadianly entrained in the cycle of a year and is independent of cold exposure and food and water availability.
Precise timing of sexual maturation is important for ground squirrels to be ready to mate in the early spring, so the offspring are born when food is plentiful and weather conditions are favorable, increasing overall survival of the species.
Our work reveals neurophysiological mechanisms of sexual maturation in 13-line ground squirrels in the face of an extreme negative energy balance during hibernation. We also found that ground squirrels use IBAs to produce sexual hormones, initiate testicular growth, and sexual maturation. This work was supported by National Institute of Health, National Science Foundation, and CAPES.